Hello everyone, welcome to our third lecture of Unit 1 of our microbiology course. In this lecture we are going to review some of the basic chemistry concepts that you'll need to understand kind of the rest of this chapter as well as specific topics in Units 2 and 3. A lot of this is probably a review from your general biology class, so I'm going to go through it a little bit quickly. However, if it's been a while since you took general biology, this may be one that you want to watch a couple of times just to make sure that you remember all of these basics. Here, of course, are your lesson objectives. There are several of them this time, but again, since this is a review, you can actually probably do several of these already. So, starting at the beginning, we will start with atoms and atomic structure. Just like cells are the basic or fundamental unit of living organisms, atoms are the fundamental or basic unit of matter. In terms of their structure, they have a nucleus that is composed of protons, which are positively charged particles, and neutrons, which are neutrally charged particles. Basically, they have no charge. And then orbiting this nucleus, there are electrons, which are negatively charged or particles. And these orbit the nucleus in what we call electron shells. Individual atoms are, of course, important because they make up matter. But from a biological perspective, most of the time we aren't interested in just individual atoms. We're more interested in how atoms combine to form larger structures called molecules. So a basic definition of a molecule is two or more atoms joined together through bonds. Now, from a chemical perspective, from a chemist's perspective, there might be a more technical definition, but for biology at this level, it is sufficient to say that molecules are two or more atoms joined together through a bond. And biologists care about bonds for two main reasons. One, bonds take atoms and join them together, so they really are the forces that are holding molecules together. And when we get enough of the right types of molecules all linked together, we can form a cell. And when we get enough cells together, we can form an organism. And then two, bonds are where the energy that living organisms need for metabolism are stored. When an organism takes in food from the environment, and this is true for humans, this is true for animals, this is true for microbes, when it takes food in from the environment, what it does is it breaks that food down into smaller and smaller pieces until the individual bonds that are holding atoms together in molecules can be broken and the energy can be extracted. So whether we're talking about a bacteria or a human, anytime an organism consumes food, one of the goals of that, or one of the reasons for doing that, is so that the energy that is holding these atoms together to create a molecule can be extracted and can be used for its own metabolic purposes. In biology, there are three main types of bonds we care a lot about. The first is the ionic bond. In ionic bonds, positively and negatively charged atoms combine and transfer electrons to each other. So just to give you an example here, kind of a classic ionic bond or ionic situation here, we have sodium, which is often positively charged. We have chlorine, which is often negatively charged. And basically what that means is sodium is missing an electron and has space for an extra electron. And chlorine is negatively charged, which means it has an extra electron and is able to give that electron away. And so what ends up happening is chlorine transfers its electron to sodium. This creates a connection between the two of them, or a bond, and sodium grabs onto that electron, chlorine hangs out, and they make what's called sodium chloride. And the bond that's holding those two atoms together is called ionic. Because two ions, the sodium ion and the chloride ion, were used to create that bond. The next type of bond that's really important to biologists are covalent bonds. Covalent bonds occur when uncharged atoms combine and share electrons with each other. So you guys have all heard of water before. You probably know the molecular formula for water off the top of your head. It's H2O. And what that means is we have one oxygen atom and two hydrogen atoms. So when that happens, when we have a hydrogen molecule or hydrogen atom that connects to an oxygen and another hydrogen atom that connects to that same oxygen atom, we end up getting water. The bonds that hold the hydrogen atoms to the oxygen atom are called covalent. So this is a covalent bond, and this is a covalent bond. These are covalent bonds because in this case, none of the atoms involved had a charge, and what they're doing is basically just sharing the electrons with each other. So oxygen is sharing an electron with this hydrogen is sharing an electron with that oxygen is sharing an electron with that hydrogen, and that hydrogen is also sharing an electron with that oxygen. And that way everyone's happy, everyone gets the electrons that they need. Now oxygen is kind of a special molecule, and we're going to get to that in just a second when we're talking about our hydrogen bonds. Hydrogen bonds occur between hydrogen and highly electronegative atoms. Typically, when we're talking about living organisms, that means oxygen, nitrogen, or phosphorus. So in individual water molecules, the bonds that hold the hydrogens to the oxygen are covalent. But to be able to get liquid water, lots of water molecules have to attach to each other. And what that means is basically you have to have a hydrogen from one water molecule interacting with the oxygen of another water molecule. This bond, the bond between the hydrogen atom of one molecule and the oxygen atom of another molecule, is called a hydrogen bond. So what we have in water is, like I said, kind of a special situation where the individual bonds that hold a water molecule together are covalent, but to hold multiple water molecules together and make something like liquid water or ice, we have hydrogen bonds that are forming. So we have a hydrogen bond between one hydrogen and an oxygen, and then I've just got another example here that I'm drawing out. We've got a hydrogen of one water molecule attaching to the oxygen of another, and that is, of course, a hydrogen bond. Now, there are some other types of bonds out there that aren't quite as important in biology. There's another type of interaction called a van der Waals interaction. Van der Waals interactions are not true bonds, but they can be important in living organisms. The way they work is basically the way magnets kind of work with each other. Van der Waals interactions are when you have molecules that are kind of pushing each other away or pulling each other close. It's almost like if you take the North Pole on two individual magnets and put it together and you kind of get that push-pull situation. That's essentially what a van der Waals interaction behaves like. But our three main bonds in biology are ionic bonds, covalent bonds, and hydrogen bonds. Molecules themselves can be very simple or they can be very complex. It can be a molecule can be as simple as having two oxygen atoms joined together or two hydrogen atoms joined together, or it could be incredibly complex where you have potentially hundreds of different atoms joined together of different types. So you could have sulfur, you could have carbon, you could have hydrogen, you could have oxygen, you could have nitrogen, phosphorus, all in one very large molecule. So they can be really simple, they can be really complex. But one thing we notice in living organisms is that oftentimes our molecules have kind of a basic structure that's like the core shape or the core structure of that molecule. And additional groups are added on. These additional groups are called functional groups. They're attached to the core, and they allow a molecule to have the same core as another molecule, but to still behave very differently in a biological environment. So when we're talking about this, you've got some examples here in the picture. If this is our core molecule here, this carbon with 3 hydrogens attached to it, then we can add different functional groups on it. And these functional groups will allow that molecule to behave very differently than the other ones. Even though the core is the same, the external atoms that make up the functional group cause molecule A to behave differently than molecule B and differently than molecule C. So what this allows for is biological organisms to have kind of a core set of molecules. They have kind of a core set of structures, but we can build very large, very differently behaving molecules based on the number and the composition of the functional groups that get added to them. This is important for lots of different reasons. One of the reasons is that it allows biological organisms to have a lot of similarities with each other, but by adjusting some of the functional groups, they can start to behave differently. The same thing goes for cells. You can have cells in one living organism where they all have kind of the same basic core molecules in them with the same core structures, but by adding and subtracting different functional groups, you get differently behaving cells from each other so that you can have differentiation. Another thing that's really key, especially when we're talking in microbiology, is when we're talking about the structure of antibiotics. So we will talk about antibiotics in depth in another chapter. But one thing I just kind of want you to keep in mind right now is that many antibiotics have kind of a core structure to them based on a core type of molecule, but they have different functional groups added to them, and this allows the molecular structure of the molecule or of the antibiotic to be different and potentially be altered and adjusted to make it work better or work differently depending on the situation. So understanding functional groups, at least at a very high or broad level, is important so we understand why maybe antibiotic A works differently than antibiotic B, even though from a molecular standpoint they are very similar. So just keep that in mind as we get into future lectures. When living organisms are going through their metabolism and just going through their cellular processes that keep them alive, they oftentimes take molecules and take atoms that they get from the environment, they break them down, and they rearrange them into the arrangement or the structure that they want them to be in. So to do this requires a couple of kind of main chemical reactions. We have synthesis reactions where things are put together. You take atom A and atom B, or molecule A and molecule B, and you link them together with some kind of bond. Decomposition, where we're taking something apart. So we have a molecule made up of A and B, and we split that bond apart and we get A and B separately. Or we can have exchange reactions, where essentially we're just moving some things around. So in this example right here, we've got A and B plus C, and then we have a chemical reaction that occurs, and basically B and C change places. So A is now bonded to C, and B is off on its own. We can have another one with AB plus CD, and B and D can change places. So that would give us A and C and B and D. And there are other types of chemical reactions, but when we look at the types overall, like the main categories of reactions that allow metabolism to function, they fall into synthesis, decomposition, and exchange. Just kind of as an example, when an organism takes in food, it typically has a series of decomposition reactions that go on to break down that food. It extracts the energy, and then maybe it will go through a series of synthesis reactions and use all of those individual atoms that it separated during the decomposition stage and rearrange them and put them into an arrangement into a new molecule that it needs inside its own cell. So these are all just general terms for different types of reactions that occur during metabolism. Aside from talking about how many atoms are in molecules and whether they're core structures or functional groups, or how they get made through synthesis, decomposition, etc. Another way to look at molecules is whether they are organic or inorganic. Now, organic molecules have carbon and hydrogen at a minimum. The minimum requirement for something to be considered organic is to have carbon and hydrogen. Now, oftentimes, but not always, oxygen is also included in that in living organisms, but at an absolute minimum, organic molecules must have carbon and hydrogen. In living organisms, all of the macromolecules, the things like carbohydrates, proteins, lipids, and nucleic acids, are all organic molecules because at a minimum, they have carbon and hydrogen. Inorganic molecules lack this combination of carbon and hydrogen. So an inorganic molecule may have carbon, or it may have hydrogen, or it may have neither, but it will not have both carbon and hydrogen. Now, inorganic molecules can be just as important to living organisms as organic molecules. Water is an example of an inorganic molecule, oxygen or O2. Carbon dioxide is an example of an inorganic molecule. And all of these are important to living organisms as well. So when we look at living organisms, one of the ways to talk about the molecules that make them up is whether or not those molecules are organic or inorganic. Organic molecules often make up the structure in living organisms, whereas inorganic molecules oftentimes serve as kind of the support for those organic molecules, or they serve as sources of electrons or ways to dispose of waste products. So both of them are equally important to living organisms, and it's just important for you to understand what we actually mean from a scientific perspective when we say organic or inorganic. Another key concept when we're talking about atoms and molecules that make up living organisms is the concept of the SPONCH elements. SPONCH stands for sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. And these 6 elements are the absolute minimum that all life forms must have, at least as far as we know. So all living organisms on the planet must have, at an absolute minimum, a source of sulfur, a source of phosphorus, a source of oxygen, a source of nitrogen, a source of carbon, and a source of hydrogen. The reason for that is these elements, the SPONCH elements, make up macromolecules. So these make up things like proteins, carbohydrates, lipids, nucleic acids. If an organism is missing any one of these 6 or multiple of these 6, it will definitely die. So all living organisms, whether they are a bacteria, whether they are a plant, whether they are an animal, they must have all of these 6 atoms in their body. Now that we've talked about everything from individual atoms and molecules to things like functional groups, to organic versus inorganic, to the SPONCH elements, it's time to just do a quick review of macromolecules. The term itself, that word macromolecule, really what it means just a basic definition is something that's a large molecule. But in biology, it has kind of a special meaning. And when you hear the word macromolecule or macromolecules, really what a biologist is referring to is one of the 4 main classes of molecules that make up cell structures in living organisms. So these macromolecules are indeed large. They are based on the SPONCH elements, and typically they have repeating subunits that are joined together. So the individual subunits themselves are called monomers. And when we string them together, they create polymers. So most of the time, what we're talking about when we talk about macromolecules in biology are these monomers that have been strung together to create polymers, which are very large molecules that play a specific role in the structure and or function of living organisms. There are four main classes of macromolecules plus an energy molecule that is very significant. And the first one that we'll talk about are the carbohydrates. Carbohydrates are sometimes referred to or abbreviated as CHO. So if you see that somewhere, that's what that means. It means carbohydrate. The monomer of a carbohydrate is the sugar or the saccharide. And when we join a bunch of them together, that creates either a disaccharide if there are two together. If there are three or more together, that creates a polysaccharide. And the polysaccharides and the saccharides themselves are typically used for energy or cell structure. So carbohydrates are used primarily for energy, but they're also used for cell structures. So as an example of energy use, you guys have all heard of glucose before. Many organisms, whether we're talking about people, whether we're talking about certain types of bacteria, they use glucose, which is a type of sugar, as a source of energy. So that's an example of how carbohydrates are used as energy for cells. But a lot of times that glucose can be strung together in very, very long strings, basically, or it can be combined with another type of individual sugar and put together in a really long string, and that can make up some kind of cell structure. For example, we can take a bunch of glucose units and join them together, and that can help make up the cell wall of certain types of algae. So that would be an example of how a carbohydrate can be used to help with cell structures. Lipids are kind of a funny class of macromolecules. Technically, they don't have just one type of individual monomer. There are a couple of different individual monomers that can combine to form lipids. Lipids are a very, very broad class of molecules, and they could play a lot of different roles in living organisms. They can be used as a source of energy storage. They can be used in cell structures. They can be used in things like signaling. They can be used in pigmentation, so giving color to something. They can be used to help repel water. And there are several other roles that they can have. So an example of energy storage is many organisms, if they have excess carbohydrates that they don't need to provide immediate energy, they will take those carbohydrates, they will rearrange them, and they will store them in the form of lipid in something like adipose tissue or some other type of fatty tissue. For cell structures, one of the classic things that they're used for in cell structures is creating the cell membrane. So when you hear of things like phospholipids, those are lipid-based molecules with a phosphate group attached, and those are absolutely key in cell structures because they help separate out the internal contents of the cell from the external environment. So really, this layer of phospholipid that creates the cell membrane is what separates life from non-life, essentially. Like I said, they can be used in cell signaling, they can be used in pigmentation, they can be used to repel water. There's a lot of different things that living organisms can do with lipids. Proteins are our third class that we will talk about. The individual subunit of proteins, the individual monomer, is the amino acid. And when we link enough of those together, we end up getting a protein. Proteins are used for lots of different things in cells, again, as well. A lot of times they're used for structure. So lots of structures within a cell that provide support will be made up of proteins. They also can be used as signaling molecules. And they can also be used as things like enzymes. So enzymes help catalyze or carry out chemical or metabolic reactions. I'm kind of running out of room, so I'm going to write over here a little bit. They can be used for cell structures. They are enzymes. Not all proteins are enzymes, but all enzymes are proteins pretty much. They can be used to signal from cell to cell. So those are just some of the things that proteins do. Our last class is the nucleic acid. The individual monomers are nucleotides, and when we join them together, we get a nucleic acid. There are two types of nucleic acids. There's RNA and DNA. So RNA stands for ribonucleic acid. DNA stands for deoxyribonucleic acid. And the nucleic acids really are the source of information that the cell uses to understand when it should produce proteins, how it should handle lipids, how it should handle carbohydrates. Basically, they are like the program language of a computer or of software. So they are the code that is used for the cell, and they tell the cell what to do, when to do it, how to respond to its environment, etc. The last molecule I have listed here is ATP. ATP stands for adenosine triphosphate. And while ATP itself is not one of the four main macromolecule classes in living organisms, it still plays a significant role, and I wanted to briefly touch on it here to kind of jog your memory. We will discuss it more in depth in Unit 2 when we talk about microbial metabolism and energy use. But just as a quick reminder, kind of the basics of ATP, ATP is the source of energy that cells use to carry out their metabolic reactions. So to be able to break down carbohydrates into smaller individual units and extract the energy from those, to be able to build up protein molecules, to express the information in nucleic acids, all of that requires ATP as an energy source to happen. So while macromolecules actually make up the cells themselves, they make up the cell structure, they help the cell function, ATP is the molecule that provides the energy for the cell to be able to do all of that. So we've been talking about macromolecules and just reviewing the different macromolecules, ATP, etc. And one thing I just want to remind you of is the reason we spend so much time talking about these is because these are the molecules that actually make up cells. When you look at a cell, which is what you're looking at right here, all of the individual components are made up of the macromolecules. So if you look at the cell membrane, it is composed of phospholipids, right? So lipids are one of the four main classes of macromolecules. And so really when we look at cell structures, when we look at how a cell is put together, how it's composed, how all of the individual components are held together and what they're made of, really we're talking about macromolecules. Cells are basically a bunch of macromolecules put together in a specific arrangement that carry out a series of chemical reactions throughout that organism's lifetime so that it can survive. So it's so important to talk about macromolecules, their composition, and their function, because these are the things that actually make up cells. So you've got the cell membrane made of lipids. Most of the organelles, or these big structures inside eukaryotic cells, have some proteins in them. They will have the nucleic acid found inside the nucleus. You will have carbohydrates in various locations that will get used to help make up the cell structure, whether that's the cell wall, or they may help support the phospholipid bilayer that's found in the cell membrane. Basically, one thing you just want to understand is that when we string enough individual subunits of the macromolecules together, we get the actual structures that are going to be used to make up the cell. And in this diagram, we're looking at the eukaryotic cell, but the same thing is true for prokaryotes as well. So we'll talk about cell structure and function in depth in the next couple of lectures, but I just wanted to help give you a visual and help you remember that macromolecules are so important because when we put them together in the right amounts and the right arrangements, we actually get cells, and cells are, of course, the basic unit of life. We are almost done with this lecture. Just a little bit more about ATP, just kind of as a review. ATP is not one of the four major classes of macromolecules, but it is a macromolecule in the sense that it is a large molecule. It's composed of 3 parts: a nucleotide called adenine, a sugar group, and 3 phosphate groups. It's created by cells during metabolic processes. The reason that it is used in cells to supply energy for those other reactions that we talked about previously, and for creating cell structures, for using cell structures, for making macromolecules or dissolving macromolecules, is because these phosphate groups store a lot of energy. So, like I said, we'll go into more detail about its creation and its use in future chapters, but I just wanted to remind you about ATP, about the fact that it exists and that cells need to be able to create it and use it to be able to create and use other macromolecules. And then finally, we'll just do a quick review of pH and osmolarity. pH basically refers to the number of free hydrogen ions in a solution. So something with a lot of free hydrogen ions is an acid. And something with very few free hydrogen ions is a base. Bases have a lot of hydroxyl ions. OH stands for hydroxyl. And the way the scale works is anything with a pH below 7 is considered an acid. Anything with a pH above 7 is considered a base. Oh, sorry. I need to switch to my pen. I keep forgetting to do that. And neutral is considered exactly 7. So something that is neutral is considered to have a pH of 7, or something that has a pH of 7 is considered neutral. Most organisms live and survive best in neutral-ish pHs. It can be a little lower than 7 or a little higher than 7 in certain circumstances or in certain locations in a body or in a cell. But overall, most living organisms tend to live right around the neutral range. Later in the semester, in subsequent chapters, we will talk about organisms that live on the extremes of the pH scale. So there are some organisms that like really acidic environments and some that like really basic environments. And we'll talk about the physiology of those and where we can find them in the environment. In terms of osmolarity, this one is sometimes not fun for a lot of people. But basically, when we're talking about osmolarity, we're talking about the relative number of solutes in a certain location. Most of the time, the location that we're talking about is the cytoplasm of a cell. So we can group solutions and cytoplasms into kind of 3 main categories. We can say hypotonic solutions, isotonic solutions, and hypertonic. Hypotonic solutions are those that have fewer solutes than the cytoplasm of a cell. So this is our cell here. It has that lipid membrane around it, and the green dots represent solutes. We can see that there are more solutes inside this cell than in the solution. So this solution is a hypotonic solution. Hypo, that prefix, means lower. So in a hypotonic solution, we have fewer solutes than inside the cell. So the solution has fewer solutes than the cell has. In an isotonic solution, we have the same number of solutes inside the cell as outside the cell. So the solution has the same number of solutes that the cytoplasm of the cell does. Iso, that one means same. That prefix means same. So same number. And then our last one is hypertonic. A hypertonic solution has more solutes outside the cell than inside the cell. So hyper, that prefix means high. And so when we talk about a hypertonic solution, we have more solutes outside the cell than inside. Sorry, I'm getting to the edge of my iPad case and it's making it hard for me to write a little bit. So one of the things that's so tricky about osmolarity and things like hypotonic, isotonic, hypertonic is that trying to understand the movement of water in relationship to all of this can be a bit challenging. If you take a cell and put it in a hypotonic environment, water will tend to go into the cell. So you take this cell, drop it into this hypotonic solution, water is going to come into the cell, and the cell is going to swell up and potentially burst. If you take a cell and you drop it into an isotonic solution, the same amount of water that moves into the cell will move out of the cell. So you have a net balance basically of zero. So the cell is not going to get larger or smaller. It's not really going to change shape. It's going to keep the same amount of water and solute balance that it had when you put it in there. In a hypertonic solution, water is going to go out of the cell into the external environment, and the cell is probably going to shrivel up and potentially die. So I think the thing that tends to trip people up a little bit is just remembering with the hyper, iso, and hypertonic, hypo, iso, and hypertonic, where the water ends up going and what will happen to the cell. So hopefully this helps remind you what will happen in those different situations.